Hello viewers, I am Titikksha, Research Scholar, Department of Zoology, Gauhati University. In this session, I shall discuss with you my topic amino acid, its structure, classification and general properties. I'd like to dedicate this video to my teachers, Jogen Chandra Kalita sir and Monidi Prasa, for giving me this opportunity to be a part of this great initiative taken by Zoological Society of Assam in order to help the students of life science during this pandemic caused by coronavirus. So at the end of this presentation, one will learn what are the building blocks of protein, importance of protein as a biomolecule, what is the structure of an amino acid, how a polypeptide chain is formed, and classification of amino acids. Now, importance of protein as a biomolecule. Proteins are the macromolecules that carry out virtually all of a cell's activities. They are the molecular tools and machines that make things happen. Proteins have shapes and surfaces that allow them to interact selectively with other molecules. Proteins, in other words, exhibit a high degree of specificity. Among the various functions of proteins act as enzymes, structural cables, as hormones, growth factors and gene activators, as membrane receptors and transporters, as contractile filaments and molecular motors. Proteins constitute the machinery for biological movements. Proteins also act as antibodies, serve as toxins, form blood clots, absorb or refract light, and transport substances from one part of the body to another. Now, if we go into the historical background of the discovery of amino acids, we know now that 20 different amino acids are commonly found in proteins. The first to be discovered was asparagine in 1806. The last of the 20 to be found, threonine, was not identified until 1938. All the amino acids have trivial or common names, in some cases derived from the source from which they were first isolated. Asparagine was first found in asparagus and glutamate in wheat gluten. Tyrosine was first isolated from cheese. Its name is hence derived from the Greek word tyros, which means cheese. And glycine, from the Greek word glykos, which means sweet, was so named because of its sweet taste. Now, what are the building blocks of proteins? Proteins are the polymers made of amino acid monomers. 20 different amino acids are commonly used in the construction of proteins, whether from a virus or a human. The structures of amino acids. Now, all amino acids have a carboxyl group and an amino group which are separated from each other by a single carbon atom, the alpha carbon. So in the figure, you can see a general structure of an amino acid. The carbon atom, which is also known as the alpha carbon, it has carboxyl group, an amino group, an hydrogen atom and an R group. Now, how a peptide bond is formed? Formation of a peptide bond occurs by the condensation of two amino acids. During the process of protein synthesis, each amino acid becomes joined to two other amino acids, forming a long, continuous, unbranched polymer called a polypeptide chain. The amino acids that make up a polypeptide chain are joined by peptide bonds that result from the linkage of the carboxyl group of one amino acid to the amino group of its neighbor with the elimination of a molecule of water. Here in the figure, there is a demonstration of a peptide bond which is formed by condensation, as you can see. The alpha amino group of one amino group acts as a nucleophile to displace the hydroxyl group of another amino acid, forming a peptide bond. As you can see, it is shaded in yellow. Now, stereoisomerism in alpha amino acid. So as we know that the central carbon atom is in an amino acid is bonded 4 different groups, that is an carboxyl group, an amino group, an R group and a hydrogen atom. Hence the alpha carbon is a chiral center. The tetrahedral arrangement of the bonding orbitals around the alpha carbon atom allow the 4 different groups to occupy 2 unique spatial arrangements, and thus amino acids have 2 possible stereoisomers. Now what is stereoisomers? That is, they're the compounds that is differing only in spatial arrangement of atoms. So here in amino acids we see there are stereoisomerism. So the compound can exist as a pair of non-superimposable mirror images of each other. The 2 forms represent a class of stereoisomers and they are known as enantiomers. All molecules with a chiral center can rotate plane polarized light and are optically active. Here in the figure, you can see there is stereoisomerism in alpha amino acids. The 2 stereoisomers of alanine, that is L and D-alanine, are shown, and they are non-superimposable mirror images of each other, or they are enantiomers. Another very important point to be noted is that nearly all biological molecules with a chiral center occur naturally in only one stereoisomeric form, that is either D or L. Now the amino acid residues in protein molecules are exclusively L stereoisomers. D amino acid residues have been found only in a few, generally small peptides, including some peptides of bacterial cell walls and certain peptide antibiotics. Now here's a chart. You can see there is abbreviations for all the amino acids. There's a 3-letter abbreviation and 1-letter abbreviation in this chart. Like in alanine, for example, 3-letter abbreviation is ALA, or you can say ALA, and 1-letter abbreviation is capital A. Similarly, arginine is ARG, and 1-letter abbreviation is capital R. So one can go through this table also. Now, classification of amino acids. Now, classification is mainly based on the character of their side chains. The side chain or R group bonded to the alpha carbon is highly variable among the 20 building blocks. And it is this variability that ultimately gives proteins their diverse structures and activities. So, amino acids fall roughly into 5 categories. The first one, that is the non-polar aliphatic R groups. What are non-polar aliphatic R groups? And the R groups in this class of amino acids are non-polar and they are hydrophobic in nature. Secondly, the side chains of alanine, valine, leucine and isoleucine. As you can see, their chemical structure has been given in the image. They tend to cluster together within proteins, stabilizing protein structure by means of hydrophobic interactions. Then glycine. Glycine, you can see in the image, it has the simplest structure, and here the R group is a simple hydrogen atom. Methionine, one of the 2 sulfur-containing amino acids, has a non-polar thioether group in its side chain. And then proline. Proline has an aliphatic side chain with a distinctive cyclic structure. Here you can see in the image. Now, the second group, that is the aromatic R groups. Phenylalanine, tyrosine and tryptophan with their aromatic side chains are relatively non-polar, or they are hydrophobic. You can see the image of phenylalanine, tyrosine and tryptophan with their aromatic side chains. Now, the third group, that is the polar uncharged R groups. Now, the R groups of these amino acids are more soluble in water or more hydrophilic than those of the non-polar amino acids because they contain functional groups that form hydrogen bonds with water. These class of amino acids include serine, threonine, cysteine, asparagine and glutamine. One can go through the chemical structure of these. Polar uncharged R groups is shown in the image. Now, fourthly, positively charged or basic R groups. Now, the most hydrophilic R groups are those that are either positively or negatively charged. So, these groups include lysine, arginine and histidine. They are positively or negatively charged and they are basic in nature. One can go through the image. Then, negatively charged acidic R groups. The two amino acids with a net negative charge are aspartate and glutamate, each of which has a second carboxyl group. So, they are negatively charged acidic R groups. As you can see their chemical structure. Now, uncommon amino acids. So, in addition to these 20 common amino acids, proteins may contain residues created by modification of common residues already incorporated in a polypeptide. Among these uncommon amino acids, you can find there is 4-hydroxyproline, a derivative of proline, and 5-hydroxylysine derived from lysine. The former is found in plant cell wall proteins and both are found in collagen, a fibrous protein of connective tissues. 6-methyllysine. It is a constituent of myosin, a contractile protein of muscle. Another important uncommon amino acid is gamma-carboxyglutamate, found in the blood clotting protein prothrombin and in certain other proteins that bind calcium as part of their biological function. More complex is desmosine, a derivative of 4 lysine residues which is found in the fibrous protein elastin. Now, classification of amino acids based on their demand for nutrition. Like they are mainly classified into essential amino acids or indispensable amino acids and non-essential amino acids. Now, amino acids whose carbon skeleton cannot be synthesized de novo in animal cells, or those that are normally or insufficiently synthesized de novo by the organism relative to its need for growth, reproduction and normal maintenance, and therefore must be supplied by the diet. They are essential amino acids. Essential amino acids include histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan and valine. Non-essential amino acids. Now, what are non-essential amino acids? Amino acids that can be made from simpler precursors and are thus not required in the diet. This include alanine, arginine, aspartate, asparagine, cysteine, glutamate, glutamine, glycine, proline, serine and tyrosine. Now, significant application of amino acids. Firstly, in artificial degradable polymers with the composition of alpha amino acids. Now, artificial polymers with the composition of alpha amino acid are very promising, as it has an innumerable use in biomedicine, particularly in the fields of tissue engineering and drug delivery. Secondly, the significant role of amino acids during pregnancy, that is their nutritional support. Now, both non-essential and essential amino acids regulate key metabolic pathways to improve health, survival, growth, development, lactation and reproduction of organisms. Some of the non-essential amino acids, example glutamine, glutamate and arginine, also play important roles in regulating gene expression, cell signaling, antioxidant responses, immunity and neurological function. Thirdly, fluorine-containing amino acids for drug design. Fluorine-containing amino acids are becoming increasingly prominent in new drugs due to two general trends in the modern pharmaceutical industry. Firstly, the growing acceptance of peptides and modified peptides as drugs. And secondly, fluorine editing has become a prevalent protocol in drug candidate optimization. So here are the references that I have gone through. Thank you to all my viewers for patiently hearing this session.